हॅलो माय डिअर फ्रेंड्स, वेलकम टू एस क्लास एज्युकेशन. आज आपण पाहतोय 11th स्टँडर्ड बायोलॉजी महाराष्ट्र बोर्डमधील चॅप्टर बायोमॉलिक्युल्सच्या एक्सरसाइजमधल्या सगळे क्वेश्चन्सचे आन्सर्स. सो लेट अस स्टार्ट. पहिला क्वेश्चन आहे चूज करेक्ट ऑप्शन. त्याच्यामध्ये पहिल्यांदा आहे ए, शुगर, अमायनो ॲसिड अँड न्यूक्लियोटाइड्स युनाइट टू देअर रेस्पेक्टिव्ह सब युनिट्स टू फॉर्म डॅश. A. Bioelements. B. Micromolecules. C. Macromolecules. And D. All of these. So correct answer आहे option C. Macromolecules. Next, glycosidic bond is found in dash. A. Disaccharides. B. Nucleosides. C. Polysaccharides. And D. All of these. So the answer is all of these. C. Amino acids in a polypeptide are joined by dash bond. A. Disulfide bond. B. Glycosidic. C. Hydrogen bond. And D. None of the above. So the correct answer is none of the above. Question D. Lipids associated with cell membrane are dash. A. Sphingomyelin. B. Isoprenoids. C. Phospholipid. D. Cholesterol. E. Linoleic and linolenic are dash acids and dash acids are referred as essential fatty acids since they cannot be synthesized by the body and hence must be included in daily diet. Option A. Arachidonic. Option B. Oleic. Option 3. Stearic. Option D. Palmitic. So correct answer is option A. Arachidonic. F. Hemoglobin is a type of dash protein which plays indispensable part in respiration. A. Simple. B. Derived. C. Conjugated. And D. Complex. So the correct answer is option C. Conjugated. G. When inorganic ions or metalloorganic molecules bind to apoenzyme, they together form dash. A. Isoenzymes. B. Holoenzymes. C. Denaturated enzyme. And D. None of the above. So the correct answer is B. Holoenzymes. Next आहे H. In enzyme kinetics, Km is equal to Vmax divided by 2. If Km value is lower, it indicates A. Enzyme has less affinity for substrate. B. Enzyme has higher affinity towards substrate. C. There will be no product formation. And D. All active sites of enzyme are saturated. So the correct answer is option B. Enzyme has higher affinity towards substrate. Next question, question number 2. Solve the following questions. A. Observe the following figure and name the type of bond shown by arrow in the structure. So the arrow is pointed here. So the answer is the figure given in double-stranded DNA and the type of bond shown in the diagram is hydrogen bond. So this is the answer. Next question. Answer the following questions. A. What are building blocks of life? Answer. Life is composed of 4 main building blocks: carbohydrate, proteins, lipids and nucleic acids. हे सगळे आन्सर्स टेक्स्टबुकमध्ये आहेत. सो तुम्ही टेक्स्टबुकमधून आन्सर्स लिहिलं तरी पण चालेल. हे सगळे आन्सर्स तुम्हाला टेक्स्टबुकमध्ये पाहायला मिळतील. बी आहे एक्सप्लेन दी पेप्टाइड बॉन्ड. Answer. A peptide bond is an amide type of covalent chemical bond that links the 2 alpha amino acids from C1 of one amino acid and N2 of another amino acid. C. How many types of polysaccharides you know? Answer. There are 2 types of polysaccharides. First, homopolysaccharide. It contains same type of monosaccharides, that is starch, glycogen, cellulose, etc. Heteropolysaccharides. It contains 2 or more different monosaccharides. For example, hyaluronic acid, heparin, hemicellulose, etc. Enlist the significance of carbohydrates. Answer. Significance of carbohydrates are as follows. First, carbohydrates supplies energy for metabolism. Second, glucose is the main substrate for ATP synthesis. Third, polysaccharide serves as a structural component of cell membrane, cell wall and reserved food as starch and glycogen. Fourth, lactose, a disaccharide present in the milk, provides energy to babies. E. What is reducing sugar? Answer. A reducing sugar that is capable of acting as a reducing agent due to presence of free aldehyde or ketone group is called a reducing sugar. These sugars reduce the Benedict reagent Cu2+ to Cu+ since they are capable of transferring hydrogens to other compounds and a process is called reduction.
मित्रांनो, अशाच पद्धतीने जर तुम्हाला एक्सरसाइजमधल्या सगळ्या प्रश्नांची उत्तरं हवी असतील तर हे जे ओसवाल पब्लिकेशनचं बुक आहे हे तुम्ही रेफर करू शकता. याच्यामध्ये एक्सरसाइजमधल्या सगळ्या प्रश्नांची उत्तरं त्यासोबतच शॉर्ट नोट्स पण पाहायला मिळतील. लिंक्स खाली डिस्क्रिप्शन बॉक्समध्ये अवेलेबल आहेत.
नेक्स्ट, what is the basic difference between saturated and unsaturated fatty acid? So saturated fats and unsaturated fats. Saturated fats are in solid phase at the room temperature. Unsaturated fats are in liquid phase at room temperature. Next, saturated fats are not get spoiled and unsaturated fats are get spoiled easily. Saturated fats are increasing the blood cholesterol level by depositing in the inner wall of arteries. Unsaturated fats are decreasing the blood cholesterol level and have many health benefits. Next, saturated fats contain single chain of carbon atoms with single bonds. Unsaturated fats contain chain of carbon atoms with one or more double or triple bonds. Saturated fats are obtained from animal fats, palm oil, etc. Unsaturated fats are obtained from plant and vegetable oils, etc. Next question G. Enlist the examples of simple protein and their significance. Answer. Examples of simple proteins are albumins and histones. Significance A. Albumin. Albumin is one of the main protein in blood. Second, albumin helps in transportation of substances like hormone and drugs in the body. Third, albumin maintains the pressure in the blood vessels. Next is histones. Histones are the chief protein of chromatin. Second, histones are involved in packaging of DNA into structural units called nucleosomes. After that, next, explain the secondary structure of protein with examples. First, the secondary structure of protein is classified in two types, alpha helix and beta pleated sheets. Second, the polypeptide chain is arranged in a spiral helix, and the two types of spiral helix are alpha helix, that is right-handed, and beta helix, that is left-handed. Third, in some proteins, two or more peptide chains are linked together by intermolecular hydrogen bonds. Such structures are called beta pleated sheets. Fourth, example of beta pleated sheets is silk fibers. Fifth, the sequence of amino acids in the polypeptide chain determines the location of its bend or fold and the position of formation of hydrogen bonds between different portions of the chain or between different chains. Thus, peptide chains form an alpha helix structure. Sixth, example of alpha helix structure is keratin. Seventh, the spiral configuration is held together by hydrogen bonds. Eighth, due to formation of hydrogen bonds, peptide chains assume a secondary structure. I. Explain the induced fit model for mode of enzyme action. Answer. In this model, enzymes are flexible structure in which the active site continuously reshapes on its interaction with the substrate until the substrate is completely bound to it. It is also the point at which the final form and the shape of enzyme is determined. So you should have to draw diagram also. Next, what is RNA and enlist types of RNA? RNA stands for ribonucleic acid. It is a long chain and single-stranded polynucleotide chain which helps in protein synthesis, functions as a messenger, and translates messages coded in DNA into protein. So there are three types of RNA: mRNA called as messenger RNA, rRNA called as ribosomal RNA, and tRNA called as transfer RNA. So as in the question, it is mentioned that you just have to enlist the types of RNA. So there is no need to explain the types of RNA. You just have to write the names of types of RNA. After that, next, describe the concept of metabolic pool. So the metabolic pool is an important concept in cellular biology because it allows one type of molecule to change into another type. For example, carbohydrates can be converted to fats and vice versa. Second, metabolic pool is the reservoir of biomolecules in the cell on which enzymes can act to produce useful products as per the need of the cell. L. How do secondary metabolites useful for mankind? Answer. Secondary metabolites are like alkaloids, nicotine, cocaine, and the terpenes, cannabinol are widely used for recreation and stimulation. Second, morphine, the first alkaloid isolated from Papaver somniferum, is used as pain reliever and cough suppressant. Third, drugs developed from secondary metabolites and it have been used to treat infectious diseases, cancer, hypertension and inflammation. Fourth, flavors of secondary metabolites improve our food preferences. Fifth, tannins are added to wines and chocolate for improving astringency. Sixth, since most secondary metabolites have antibiotic property, they are also used as food preservatives. Seventh, glucosinolates is a secondary metabolite which is naturally present in cabbage, imparts a characteristic flavor and aroma because of nitrogen and sulfur containing chemicals. It also offers protection to these plants from many pests. Solve the following questions. So you have to complete the chart. Proteins and their physiological role. First one is collagen. Physiological role is to provide strength and plays structural role. Myosin, responsible for muscle contraction. Immunoglobulin IgG protects the body from infection. Hemoglobin, significant in respiration. Fibrinogen, responsible for normal clotting of blood. Next question. Answer the questions with reference to the following figure. So you can see the figure and the questions are as follows. Name the type of bond formed between two polypeptides. So here you can see two polypeptides and you have to tell the name of the bond. So the answer is disulfide bond. So here SS, you can see the bond between SS. So we can say that there is a disulfide bond. After that, next, which amino acid is involved in the formation of such bond? So the answer is cysteine. Next, amongst first, second, third and fourth structural level of protein, which level of structure includes such bond? So the answer is tertiary structure. Next, match the following atoms given in column 1 and 2. So here I have marked them with the same color. Like for RNA, the answer is uracil. For yam plant, the answer is anti-fertility pills. Koshland, the answer is induced feed model. Omega-3 fatty acids, the answer is flax seeds, and sucrase, the answer is hydrolase. So if you want, you can just take out the screenshot. Next question, what are biomolecules and explain the building blocks of life? Biomolecules are essential substances produced by our body which are necessary for life. The building blocks of life are carbohydrates, lipids, proteins, and nucleic acids. First one is carbohydrates. Carbohydrates are biomolecules made from carbon, hydrogen and oxygen. B. The general formula of carbohydrates is CH2ON n times. C. They contain hydrogen and oxygen in the same ratio as in water 2:1. Carbohydrates can be broken down to release energy. E. Based on sugar units, carbohydrates are classified into three types: monosaccharides, disaccharides and polysaccharides. Second, lipids. These are group of substrate with greasy consistency with long hydrocarbon chain containing carbon, hydrogen and oxygen. B. In lipids, hydrogen to oxygen ratio, here it should be ratio, n should not be there. Ratio is greater than 2:1. Lipid is a broader term used for fatty acids and their derivatives. They are soluble in organic solvents, that is, they are non-polar solvents. Fatty acids are organic acid which are composed of hydrocarbon chain ending in carboxyl group, that is COOH, and these are divided into saturated fatty acids and unsaturated fatty acids. Fatty acids are basic molecules which form different kinds of lipids. Different kinds of lipids are simple lipids, compound lipids, derived lipids, etc. Proteins. Proteins are large molecules containing amino acid units ranging from 100 to 3000. They have higher molecular weight. In proteins, amino acids are linked together by peptide bonds which join the carboxyl group of one amino acid residue to the amino group of another residue. A protein molecule consists of one or more polypeptide chains. Proteins contain any or all 20 naturally occurring amino acid types. Proteins have different structures like primary, secondary, tertiary, and quaternary structure. Proteins are classified into three types. Simple proteins. Simple proteins on hydrolysis yield only amino acids, for example histones and albumins. Conjugated proteins. It consists of simple protein united with some non-protein substances, for example hemoglobin. Derived proteins. These proteins are not found in nature as such, but are derived from native protein molecules on hydrolysis, for example metaproteins, peptones, etc. Next is nucleic acids. So nucleic acids are macromolecules composed of many small units or monomers called as nucleotides. Each nucleotide is formed of three components, that is pentose sugar, a nitrogen base, and a that is phosphoric acid. When sugar combined with nitrogenous base, it forms nucleoside. Nucleotide can be called as nucleoside phosphate. There are two types of nucleic acid, that is DNA and RNA. DNA stands for deoxyribonucleic acid, which is a genetic material of a cell. It is double-stranded helix, and each strand of helix is made up of deoxyribose nucleotide. RNA is a single-stranded structure having fewer nucleotides as compared to DNA. The strands may be straight or variously folded upon itself. It is made up of nucleotides. Next, explain the classes of carbohydrates with example. Based on the number of sugar units, carbohydrates are classified into three types, namely monosaccharides, disaccharides, and polysaccharides. Monosaccharides. A. Monosaccharides are the simplest sugars having crystalline structure, sweet taste, and soluble in water. Depending upon the marking scheme of the question, you can reduce the points as per your choice. They cannot be further hydrolyzed into smaller molecules. They are the building blocks or monomers of complex hydrocarbons or carbohydrates. Next, they have the general molecular formula CH2O n times, where n can be 3, 4, 5, 6, and 7. They can be classified as triose, tetrose, pentose, etc. Monosaccharides containing the aldehyde group are classified as aldose, for example glucose, xylose, and those with a ketone group are classified as ketose, for example ribulose, fructose. Next is disaccharides. Disaccharides is formed when two monosaccharides react by condensation reaction releasing a water molecule. This process requires energy. A glycosidic bond forms and holds the two monosaccharide units together. Sucrose, lactose, and maltose are examples of disaccharides. Sucrose is a non-reducing sugar since it lacks free aldehyde or ketone group. Lactose and maltose are reducing sugars. Lactose also exhibit in beta form, which is made up of beta-galactose and beta-glucose. Disaccharides are soluble in water, but they are too big to pass through the cell membrane by diffusion. Next is polysaccharides. Monosaccharides can undergo a series of condensation reaction, adding one unit after the other to the chain till a very large molecule is formed. This is called polymerization. Polysaccharides are broken down by hydrolysis into monosaccharides. The properties of a polysaccharide molecule depends on its length, branching, folding, and coiling. For example, starch, glycogen, cellulose, etc. Describe the types of lipids and mention their biological significance. Lipids are classified into three main types. First, simple lipids. A. These are esters of fatty acids with various alcohols. Fats and waxes are simple lipids. Fats are esters of fatty acids with glycerol. Molecular formula CH2 bond OH bond CH bond OH bond CH2 bond OH. Triglycerides. Triglycerides are three molecules of fatty acids and one molecule of glycerol. Unsaturated fats are liquid at room temperature and are called oils. Unsaturated fatty acids are hydrogenated to produce fats, for example vanaspati ghee. Biological significance. Fats are a nutritional source with high calorific value and they act as reserved food materials. Again, depending upon the marking scheme of the question, you can deduce the points which are present in this answer. In plants, fat is stored in seeds to nourish embryo during germination. In animals, fat is stored in the epidocytes of the adipose tissue. Fats deposited in subcutaneous tissue act as an insulator and minimize loss of body heat. Fats deposited around the internal organs act as cushions to absorb mechanical shocks. Wax is another example of simple lipid. They are esters of long-chain fatty acids with long-chain alcohols. They are found in the blood, gonads, and sebaceous gland of the skin. Waxes are not as readily hydrolyzed as fats. They are solid at ordinary temperature. Waxes from water-insoluble coating on hair and skin in animals. Waxes from an outer coating on stems, leaves, and fruits. Compound lipids. These are esters of fatty acids containing other groups like phosphates, that is phospholipids, sugar, that is glycolipids, etc. They contain a molecule of glycerol, two molecules of fatty acid, and a phosphate group or simple sugar. Some phospholipids such as lecithin also have a nitrogenous compound attached to the phosphate group. Phospholipids have both hydrophilic polar groups, that is phosphate and nitrogenous group, and hydrophobic nonpolar groups, that is hydrocarbon chain of fatty acid. Glycolipids contain glycerol, fatty acids, simple sugars such as galactose. They are also called cerebrosides, etc. Biological significance. Phospholipids contribute in the formation of cell membrane. Large amount of glycolipids and are found in the brain white matter and myelin sheath. Next, D. Explain the chemical nature, structure and role of phospholipids in biological membrane. Phospholipids have both hydrophilic polar groups and hydrophobic nonpolar groups. Phospholipids contribute in the formation of cell membrane. Glycolipids contain glycerol, fatty acids, simple sugars such as galactose and nitrogenous base. They are also called cerebrosides. Large amount of them have been found in the brain, white matter and myelin sheath. E. Describe classes of proteins with their importance. Answer. Proteins are classified into three categories. A. Simple protein. Simple proteins may be soluble in water and these are soluble in one or many more solvents. Third, simple proteins on hydrolysis yield only amino acid. Histone proteins of nuclear proteins are soluble in water. Globular molecules of histones are not coagulated by heat. Albumins are also soluble in water but they get coagulated on heating. Albumins are widely distributed, for example, egg albumin, serum albumin and legumelin of pulses are albumin. Importance. They are involved in structural component. They also act as a storage protein. B. Conjugated proteins. Conjugated proteins consist of a simple protein united with some non-protein substances. The non-protein group is called prosthetic group, for example hemoglobin. In hemoglobin, globin is the protein and the iron-containing pigment heme is the prosthetic group. Similarly, nucleoprotein have nucleic acids as prosthetic group. Proteins are classified as glycoproteins and mucoproteins. Mucoproteins are carbohydrate protein complexes, for example mucin of saliva and heparin of blood. Lipoproteins are lipid proteins complexes, for example conjugate protein found in brain, plasma membrane, milk, etc. Importance. They are involved in structural components of cell membrane and organelles. They also act as a transporter. C. Derived proteins. These proteins are derived from native protein molecules on hydrolysis. These proteins are not found in nature. Metaproteins, peptones are derived proteins. Importance. They act as a precursor for many molecules which are essential for life. What are enzymes and how are they classified? Mention examples of each class. So, classification of enzymes. Oxidoreductase. These are enzymes catalyzing oxidation and reduction reaction by the transfer of hydrogen and or oxygen. For example, alcohol dehydrogenase. Alcohol plus NAD plus in the presence of alcohol dehydrogenase will give aldehyde plus NADH2. Whenever you are explaining the types of enzymes or classification of enzymes, you should have to write the reaction. Next, transferase. These enzymes catalyze the transfer of certain groups between two molecules, for example glucokinase reaction. Glucose plus ATP in the presence of glucokinase will give glucose 6-phosphate plus ADP. Hydrolysis. These are enzymes catalyze hydrolytic reactions. This class includes amylase, protease, lipases, etc. For example, sucrase. Sucrose in the presence of sucrase will break into glucose plus fructose. Next, lyase. These enzymes are involved in elimination reaction resulting in the removal of a group of atoms from substrate molecule to leave a double bond. It includes aldolases, decarboxylases and dehydratase. Example, fumarate hydratase. Histidine in the presence of histidine decarboxylase will give histamine plus carbon dioxide. Isomerase. These enzymes catalyze structural rearrangement within a molecule. Their nomenclature is based on the type of isomerism. Thus, these enzymes are identified as resmerases, epimerases, isomerases, mutases. For example, xylose isomerase. Glucose 6-phosphate in the presence of isomerase will get converted into fructose 6-phosphate. Ligases or syntheses. These are the enzymes which catalyze the covalent linkage of the molecules utilizing the energy obtained from hydrolysis of an energy-rich compound So we are continuing the next part of this exercise in the next video. So we will restart from this question. Thank you so much for watching this video. I hope की तुम्हाला सगळ्यांना हा व्हिडिओ आवडला असेल. बाय बाय.